My name is Anna Osterholtz. I'm a PhD candidate at the University of Nevada, Las Vegas. Okay. So the title of the presentation is actually Mortuary Patterns in the Tell Abraq Tomb Assemblage. And this is regarding work on a skeletal assemblage that we have at UNLV. The tomb at Tell Abraq was discovered in 1989 and excavated in 1993 and 1998. The tomb dates to the late Bronze Age, circa 2100 to 2000 B.C.E. The tomb is associated with a large mud brick fortress. The tomb is of Umanar type, a circular above-ground tomb with two internal chambers separated by a partition wall. The recently deceased were placed in with the remains of earlier burials, leading to disarticulation and commingling of all of the individuals. It's also possible that the tomb acted as a repository for secondary burials that were processed elsewhere. The commingling may indicate a change in status of the dead as they became part of a collective of ancestors. Because of the commingling, any secondary processing or retention of elements may be difficult to identify. My role on this project really started with the identification of baseline data. So the identification of how many individuals were actually in the tomb, how many males, how many females, what was the age distribution, and all of that. And when you have a commingled assemblage, that can be actually a very difficult thing to figure out. So we started with actually a rough sort, fine sort method. So all of the left femora went in one box, all the right femora in another. And then we started to differentiate them based on age and sex from there, which ultimately led to us having a separate area in the storeroom for adults and children, and then they were boxed by element and then bagged within those boxes by sex and age wherever we could. Why is it important to actually develop this baseline data? First of all, because we actually scored individual features on every element, it allowed us to identify what was present but what was also not present. So it allowed us to identify underrepresentation of elements and overrepresentation of elements, which in commingled assemblages can allow you to actually look at the depositional patterns and identify how an assemblage was made. So that was essentially my role in it. The first thing I did was actually determine the minimum number of individuals. This is it's referred to as MNI. And this will include information about sex and age as much as possible. I'm only discussing the adults for the purposes of this presentation, or discussion, whatever. So we used a modification of Nusselt's zonal method for recordation of fragmentary remains. What this means is that it is essentially a faunal approach to looking at various parts of the bones to determine what is present and what is not present. And for us, we were interested in areas of differing bone density because it's a natural assemblage. It's not that you're not seeing intentional fragmentation. So we actually went through and scored individual features on each bone. So we used a modification of Nusselt's zonal method for recordation of fragmentary remains. Different research questions led to different analytical approaches. For the work that was done by Stoddard and Osterholtz, we used a variation of the zonal method that targeted at taphonomic indicators of violence, such as high fragmentation levels and intentional fragmentation and breakage. For Tell Abraq assemblage, we weren't as interested in intentional breakage, but far more in the degree to which bone density impacted natural attrition in an assemblage of a communal cemetery. So essentially, we were looking at how the assemblage was impacted by the continued use of the tomb. So we looked at varying skeletal features with differing densities to record what was present and what had not survived to modern times. We began with an initial sort of elements side by side. The elements were examined and recorded using a visual recording form, like the pelvis that you see. This was done for several reasons. First, even though single analysts completed the recordation of a single element, i.e., the person who started the scapula finished the scapula, the visual form allowed for consistency in when a feature was scored as present versus absent. The database linked the original accession number to the locational data collected in the field as well as the gathered baseline data. So the accession number, which is a very important kind of tracking mechanism for us in our database, will ultimately also allow for what we hope to be a 3D map of the tomb assemblage itself. The methodology was specifically designed to perform several functions. First and foremost, baseline data needed to be recorded for use in secondary analyses. Metric sex techniques were used that had at least a 70% accuracy rate, and this is better than flipping a coin. We understand that there are always going to be problems in dealing with an assemblage that is not, or dealing with a reference assemblage that is radically different from your own, the one that you're studying. However, we used the methodology that we had. We broke down the MNI based on region and element without regard to sex in this diagram. Only adults are pictured, and this diagram also shows the strict MNI, which is basically just the replication of features on the bones themselves. The features chosen show a variety of bone densities, from areas more dense, such as the talus, to those that are less dense and in other ways more fragile, such as the scapular blade. Taking the ilium as an example, the MNI for the iliac blade is a maximum of 67, while the ischial tuberosity is 104. The more dense bone is preserving better. This suggests that the less dense bone is more likely to be broken during multiple reuses of the two. The element with the highest MNI is the talus, with a minimum of 278 individuals, and again these are just the adults. And the cranial MNI is 72 based on the glabella, or this part right there on the forehead. So then the next few slides will actually break things down by sex and by age. The pubic symphysis gives us our most precise breakdown by age category. For this we used Bijkstra and Ubelacker's standards of data collection age categories of young adult, 18 to 35, middle adult, 35 to 50, and old adult, or 50 plus years. Of the 48 pubic symphyses suitable for estimation of age at death, 2 could only be classified as adult based on taphonomic damage. Of these, 1 could be estimated as a male. The other is unidentified. 23 young adults were found, consisting of 2 females, 15 males, and 6 indeterminate individuals. 21 middle adults are present, with 3 females, 15 males, and 6 indeterminates, and both of the older adults are male. The glabella also has the added benefit of being sexually dimorphic, and so the sex ratios could be determined for this element as well as the MNI. For a total of 72 glabellas, 30 could be estimated as female, 34 estimated as male, and 4 individuals were ambiguous, meaning that they scored somewhere between the males and females. This is, and 4 actually could not be used to estimate sex, usually due to missing cortical bone in the area. The feature was present enough to be counted, but not complete enough to use in the estimation of sex. So I'm going to compare these sex ratios in just a little bit. Looking at the os coxa breakdown by sex, another sexually dimorphic element is the os coxa, or pelvis. The ischial surface of the acetabulum provided the highest MNI for this element. This was broken down by sex into 11 females, making up 38% of the sexable elements, 17 males, making up 59% of the sexable elements, and 1 ambiguous individual, or making up 3%. This breakdown is roughly 60 to 70% male and 30 to 40% female, and this is going to become a fairly standard ratio for the rest of the postcranial elements. The next slide, we're looking at the elements estimated where sex is estimated metrically. For the purposes of this slide, the indeterminates have been left out. Because sex is estimated metrically for these elements, incomplete elements were often not usable were most often indeterminate. But these elements show a consistent pattern of 60 to 70% male and 30 to 40% female. This pattern agrees with that seen on the pelvis, where the sex is estimated more morphologically as opposed to metrically, because metric elements are going to be based on analysis of known samples that are radically different in terms of their geographical location and historical time period than what we're looking at for the Bronze Age UAE. Again, we understand there are problems with this, but we use what we've got. Looking at the metric versus morphological sex breakdown, we see the same consistency between morphological identifiers of sex and metric identifiers of sex. It starts to become a little bit more interesting when we look at the cranial versus postcranial sex ratios. The cranial sex ratio is radically different from what we're seeing in the postcranial elements. Now, if you look at the diagrams, you can see that males are making up a little bit more than half of the cranial sex ratio, while in the postcranial sex ratios, they're making up somewhere again between 60 and 70%. We're not entirely sure why this is, but I have a few ideas that we'll talk about in a minute. Are there missing male heads? The methodology that allows us to examine the elements for completion and for presence also let us know what was not there. And what we ended up finding was that there seemed to be a lower percentage of male crania. Now, the comparison of sex ratios between the cranial and postcranial elements is fairly clear, and we do have consistent postcranial sex ratios that don't match up with the cranium. So when examined in light of the overall MNI, this patterning is suggestive of a removal of male heads at some point during the mortuary process. The underrepresentation of male crania is an interesting pattern if, in fact, it is a real occurrence. There are several possibilities for this pattern. First, it could be a result of sampling strategy. There's a large area of damage to the tomb wall where it could be that if the crania had been actually recovered from this area, the sex ratios would appear to be far more normal for the cranium. Second, there could be a secondary deposition pit at Tell Abraq that has not yet been excavated. If this is the case, then those male crania could be in there. There are also some analytical issues with inter-observer error and sex estimation, which could be at play here. If the pattern does exist, however, it brings up some interesting ideas about ancestor veneration, including an extended mortuary ritual. Lamps, ceramics, and feeding shells have been found within the tomb, suggesting possible feasting or food offerings placed in with the interments. So the retention of heads has not been documented for Umanar assemblages before, but the retention of heads is well known in other parts of the world, dating back to at least the Neolithic in Europe and the ancient Near East. The comparison of other Umanar tombs' mortuary patterning is difficult for multiple reasons. First, there's a general understanding that variability in Umanar tomb construction and use is incredibly high. Second, Tell Abraq lacks any associated ossuary subsurface pits for secondary burial. At sites such as Hilli and Ras Al Jins, these pits acted as repositories for remains that were typically processed within the tomb structure itself, and so they'll show a different distribution. And what you're seeing here on the left is actually a diagram showing the different representation of these elements within different pits in Munoz's analysis. And what you see is the larger an element appears in that diagram, the more prevalent it is in the assemblage inside that pit. So the tomb itself shows an overabundance of hand and foot bones, while some of the pits show an overabundance of crania and an overabundance of long bones. So what that tells you is that the patterning of disposal after processing is specific to the pits themselves. We don't see that pattern at Tell Abraq. We see a general representativeness of all of the elements of the body are fairly consistent. So we know that we have secondary processing and deposition within the same structure, which is a very different pattern from what we're seeing at other Umanar tombs. Finally, to our knowledge, this is the first feature-based approach that takes a very detail-oriented view of determining the MNI for the assemblage itself. And until we have more feature-based approaches like this and have a more detail-oriented idea of looking at individual features on the bone as opposed to the bones themselves, it will be very difficult to compare what we're seeing at Tell Abraq to other Umanar assemblages. So that's it.